Thank you, Bernd. And then our final speaker is Alison Webster from GSK.
Thank you very much. It's a great pleasure to be here and to be able to present this on behalf of GSK and my colleagues in the Disease of the Developing World Discovery Performance Unit, quite a mouthful, based in Tres Cantos in Spain. So I'm really going to spend most of the time talking about our more advanced projects, but I think it's helpful just to have an overview of our portfolio, looking at our early phase projects in the hit stage and lead optimization. I'll stand a bit closer, sorry. So we have target-based screening going on, and we have programs looking at cell wall inhibitors. We also have a DPRE1 inhibitor, CASA as well. We are also doing whole cell screening, looking at replicating TB and non-replicating TB, with the aim of identifying drugs that will have sterilizing properties. Also, we are interested in the concept of using beta-lactams in TB, a class of drugs that essentially really been understudied in TB, and that's the topic of a collaboration that we have going, which I'll talk a little bit more about in due course. We also have a program looking at inhibitors that are active in the presence of cholesterol, the putative mechanism of action being that if cholesterol is the carbon source, it causes accumulation of toxic cholesterol metabolites. And this can be seen if you put cholesterol into the in vitro systems, or indeed if you actually look at intracellular models of TB. So that's a very interesting program as well. We're also looking at ethionamide boosters, quite an interesting concept, to really improve the activation of ethionamide to its active form so that lower doses of ethionamide can be used and therefore avoid the potential toxic effects of ethionamide and its tolerability. So that's a project that we have going as well in that lead optimization stage. We also have programs in tryptophan synthetase inhibition as well. And the reason that I'll talk about the later projects a little bit as well, the antibiotic consortium, which we're just embarking on. We also have a project looking at inhibition of IL-4. So this is a host-directed therapy. It's really a proof of concept study in a small number of subjects, just testing the hypothesis whether manipulating this particular target of IL-4 actually could produce some benefits in terms of the host immune response, switching between Th1 and Th2 to have a better immune response to the disease. The LURS inhibitor, I'll talk a little bit more about in a moment. But I just wanted to refer to the color coding, which actually indicates all the various collaborations that we work in. And this is very much a theme in our work at GSK, is to work through consortia and collaboration, through which we get access to great expertise and resources. And also the governance of these collaborations, I think, gives independent peer review and validation of our scientific approach. So for all of those reasons, we find that a very, very valuable way of working. So now just turning to our more advanced projects. As I've mentioned, we're focused on the discovery of new clinical candidates for multi-drug resistant TB. We have in-house expertise on novel imaging techniques and animal models. And the source of projects, as I've indicated is through phenotypic hits, but also through target-based mechanism of action. So two big activities that we're embarking on. The first is a new chemical entity for TB, a new oral low-dose drug for TB, which will be moving to first time in man very shortly. And the second is the repositioning of beta-lactams for MDR-TB. As I mentioned, beta-lactams have been understudied, but I think there are a lot of reasons to be interested in beta-lactams as potential agents. Through their mechanism of action on the cell wall, they are rapidly cytocidal. They have a huge safety database in terms of being, in general, safe and well tolerated. They've been widely used in pediatrics for very common indications. So I think there's wealth of evidence supporting the safety profile in general for beta-lactams. So our new molecule that I was just alluding to, GSK-070, it's a potent, selective anti-tubercular with low MICs and selective for TB, inactive against other bacteria. It's active in our acute, in vivo, acute and chronic models. The the middle graph there shows the activity in the acute murine model, achieving more than our desired 2-log reduction in the acute mouse model. Similarly, in the chronic marmoset model, we're seeing good activity too. So good evidence to move forward with this project. Projected low human dose, it's actually a very potent molecule, as I mentioned. We've completed our GLP tox package with a therapeutic index that supports moving forward. And we have ongoing studies at the moment looking at how to best partner this drug in terms of other novel agents in development, looking at combination studies, both to identify partners that reduce the bacterial load most effectively, but also looking at those combinations that have the biggest effect in preventing relapse through our partnership with Johns Hopkins. So just returning to the topic of beta-lactams, this was published in the New England Journal, demonstrating the proof of concept that in an early bactericidal activity clinical trial, that meropenem in combination with amoxiclav, so that's amoxicillin and clavulanate, actually has a demonstrable effect in reducing bacterial load in the sputum. And you'll see there that it actually compares pretty well with the standard combination HRZE. So the objectives for the antibiotic consortium that we're just embarking on is primarily to renew the pipeline for anti-TB antibiotics. So establishing the proof of concept for our new oxaborazole drug, 070, looking at repurposing marketed beta-lactam antibiotics, and identifying a combination of beta-lactams that would be suitable as an oral, preferably, or as a minimum once daily intravenous or intramuscular application, and incorporate that best beta-lactam combination into an explorative salvage regimen for patients with few treatment options with XDR-TB. Other objectives for the consortium are to evaluate the use of clinical imaging, such as PET, and also looking at biomarkers as potential surrogates or as early indicators of activity, which will give us confidence for moving forward into longer-term drug combination trials. And the last objective is really to build on the good work that's already been commenced in terms of mathematical modelling to better understand the potentially rich data that we're going to derive from these more intensive monitoring of patients, and help to tie that back to what we see in preclinical models so that we can really better develop drugs in future. So this is the kind of consortium in a very brief overview. The phase 2A EBA of our new oxaborole molecule, the beta-lactam antibiotic combinations that we're going to test in EBA, and the XDR-TB combo, which would be an observational study, and using data from all of those studies to evaluate and identify biomarkers and improve our modelling of the disease. And this work will start with the first EBA studies with the beta-lactam antibiotics very shortly. And just finally, to finish on our collaborators in that particular consortium, the University of Cape Town and TASC, also based in South Africa, who will be doing the EBA studies, University of Tromsø, doing much of the modelling, the research centre in Borstal, who will be looking at the XDR salvage regimen, and GlaxoSmithKline in Triskantos. Thank you very much.